Welcome, digital pathology trailblazers, live from SITC.
Learn about the newest digital pathology trends in science and industry. Meet the most interesting people in the niche and gain insights relevant to your own projects. Here is where pathology meets computer science. You are listening to the Digital Pathology Podcast with your host, Dr. Alexandra Zhurov.
And I have special guests today, David from Biocare and Don from Hamamatsu. Thank you so much for joining us live. Let's start with you, Don. Who are you at Hamamatsu and why are you here?
Yeah, so my name is Don, and I lead the strategy and business development for Hamamatsu for Multiplex platform. So Hamamatsu is here showcasing at SITC mainly because we have a product targeting multiplex immunofluorescence. And SITC is all about understanding the immune profile of a patient or of a translational study. So we are here to showcase our product and demonstrate and also work with our partners and meet new people.
We're going to talk about this partnership in a second. So, SITC, the Society for Immunotherapy and Cancer, this is the 40th anniversary of this conference. So, I bet there's going to be some special things in addition to the special talk that is co-sponsored by Hamamatsu and Biocare. I want to invite you immediately into it tomorrow at 12:30. If you're here at SITC, drop a comment. And first of all, visit us at our booth. Currently, we're at 4:15, and I think the exhibit hall opens at 10 officially, right? We're unofficial yet. So tomorrow talk at 12:30. I'm going to tell you about it in a second. But now, David, a couple of words about you and Biocare's role here at this conference.
Yeah, absolutely. Thanks, Dr. Alex. My name is David Anderson. I oversee business development and strategic partnerships for Biocare Medical. I'm here with our life sciences team who are primarily focused on supporting our pharma, biotech, and CRO partners. And we're showcasing our Encore Pro X multiplex immunofluorescence staining instrument. So this is an IHC and immunofluorescence staining box that's compatible with a variety of different spatial biology assays and ultimately feeds digital or slides for digital imaging into multiplex immunofluorescence scanning instruments like the Multiplex, which we are so excited to be supporting here at SITC now and moving forward with our partnership with Hamamatsu.
So yeah, maybe let's talk about the partnership. When
Sure.
did you guys start to partner? Why did you decide to partner? And then we're going to dive a little bit into the trends, what you have
Sure.
observed in immune oncology and multiplexing. But let's talk about the partnership first. Don.
This partnership started exactly a year ago.
1 year
It was
ago.
Last year at SITC.
You guys can talk. And
David and I
listen.
met and we were trying to understand. So if you look at multiplex immunofluorescence, the whole premise is we have to reduce the entry to barriers so people or the researchers can actually do their work. They should not have to tinker with what's data, what's scanner, or any of those issues. So we want to simplify that process. So initial discussions we started last year, and we have been going through several validation studies to make sure that our protocols we are able to utilize the slides which were stained using Biocare platform and able to image it on Hamamatsu. So we are in a position right now to publicly announce these partnerships and how we are working together. So the talk tomorrow will focus on some of those data at the second part of the discussions.
So I want to highlight something. What I want to highlight here is the necessity for this kind of partnerships. Exactly what you said, to lower the barrier to entry, because that kind of ties into the perception in the diagnostic world, and we are going to be debunking these myths tomorrow, because the talk tomorrow, let me the title is going to be about the clinical utility, right? So like the perception of multiplexing and immunofluorescence is, oh, it's difficult, it's not that useful in the clinic. And and it's definitely more difficult than H&E, right? But the value that it brings and its utility has been proven. So, David, a couple of words from you about the partnership.
Absolutely. So there's so many pieces of the puzzle in this workflow, right? From the antibody selection, the panels that are developed by the spatial biology assay providers, the staining instrumentation, scanning, and down into analysis. And while there's some organizations that have multiple pieces of the puzzle, not all have all of them. And Biocare as an organization is ultimately a staining provider. We don't have digital slide scanners in our portfolio, so we were very actively looking for a partnership with the best of the best when it comes to multiplex immunofluorescence scanning. So we wanted to complete a piece of our puzzle and be able to simplify that process, bring the Encore Pro X and all of the strengths that it brings with all the spatial biology assays it's compatible with into the digital space. And Hamamatsu was the ideal partner to do that with. Moreover, the Multiplex is just a phenomenal instrument. As Don said, we've done a number of validation studies with our partners across the United States, and the data coming off their instrument showcasing what Encore Pro X can do is just absolutely phenomenal. So we're excited to get the partnership off the ground. There's a lot of great things that are coming, which we'll be highlighting in the spotlight session tomorrow and into 2026. But that's really where it came from, is merging two organizations that have great pieces of the puzzle to do more together than we could individually.
Okay, so we have two pieces of the puzzle.
Yep.
We have the staining, we have the scanning. Let's talk about the trends. Like, are you seeing any changes? Are you seeing a change also in perception and in understanding of the technology to profile to more microenvironment? Is there an uptake, or are we still like trying to push it? And what do you think?
So I think it's there are two different elements to it. One side is the research side, where people want to look at 30, 40, 50 biomarkers. So that's purely on the research side. But if you look at translational space, people are coming to this conclusion right now. All these high plex has to be consolidated to a smaller, actionable, smaller plex. So they could do large scale studies in the translational space. Like Carlos, Dr. Bifulco's talk tomorrow, where he will talk about a panel which we developed, where he was able to do some retrospective study with 5 counts, because those are actionable steps people are able to take it. But in general, I believe, or how much that we believe, simplifying the process will enable people to utilize these technologies beyond what they are doing it in a research environment. That's why this partnership with Biocare is very important for us, because it simplifies one step. Like David said, that there is multiple pieces to this puzzle. This is just one piece. We're in the process of building that entire workflow, and we are pretty optimistic we could help bridge that gap as we move forward.
So I like to I don't know. I like to compare life to a funnel and the result, and in that, well, not ideal, but basically the funnel that I see is like the research with a lot of markers that you want to visualize. Then you narrow them down, and I see this like drive to narrow it down even more to a bright field IHC.
Yeah, right.
It's not always possible. Where is there a balance? Have you seen projects, collaborations where actually it had to stay at the multiplex level? Like, what do you see at Biocare?
Yeah, so I think we are primarily a chromogenic IHC provider when we are very much involved in the multiplex immunofluorescence space. Inherently, chromogenic and maybe duplex or triplex IHC will have its limitations, right? Now, we'd like to simplify down to those individual markers, but inherently, when you're dealing with immunotherapy, tumor microenvironment, there's very complex biology at play, right? You can't maybe limit that to 2 or 3 markers when you're dealing with therapeutic response or looking at it in some sort of predictive or prognostic setting. And so I think multiplex immunofluorescence absolutely has its place, but to Don's point, finding the right plex, right? It's not necessarily high plex, but it's right plex that matters. And that 4, 6, 8 is going to be absolutely critical as we look toward the potential of clinical clinical or therapeutic application. So in the moment, can't necessarily cite an application or a publication that finds that sweet spot. But I think in general,
talk tomorrow.
Thank you. Yes,
yeah.
I think the trend is absolutely there. That sweet spot in sort of mid-single digit plex is the place to be. And multiplex immunofluorescence is critical for that, and digitization is critical for that, right? There are limitations to the human eye. We have to digitize. You have to use digital imaging to do exactly what we're speaking about for these applications. So.
Very much. I want to stay a little bit longer on the markers because
sure.
When, let's say, somebody's exploring this space, they already have some idea on the research side what they want. When they come to you, what do they ask for? Like, do they ask for kids? Do they have like a wish list? Oh, all these markers together.
Yeah.
How do you deal with that?
So this is one I'm actually going to pass back to Dawn, so because he may be closer to this one here. But what I will say right now, and it's a plug for some of our additional partnerships, Biocare is not an assay provider, right? We have a staining platform, but we've partnered with incredible organizations that have these spatial biology assays. So, Akoya Biosciences, Cell Signaling Technologies, Ultivue, right? These are the organizations that are using antibodies to create the actual panels and the assays that are going onto our instrument to be stained and then ultimately go on to the Hamamatsu box. So we absolutely are getting questions around panels. We have a phenomenal life science team that guides customers and researchers to either the right provider or the right solutions to answer exactly that question. But Biocare doesn't make those recommendations. That's where we rely on the partnerships and the spatial biology community to help guide researchers to those decisions. But Don, you might be able to answer that better.
You guys make recommendations. Feel free to make recommendations.
Yeah, I wish I can make recommendations, but
no, you can totally recommend the partners that you decided to partner with.
So this particular study tomorrow with Dr. Bifulko presenting, that particular panel was developed with Ultivue. So these are assay providers who can actually build these panels.
Yep.
But as we decide, as we move further along, there is a need for organizations such as Hamamatsu or Biocare to work very close partners and develop some pre-built panels or pre-built assays that people could use. So I think that's something organizations such as us will need to take that burden so that adoption can happen better. But it's a process. We just have to go through the process as we move along.
I'll add maybe one other comment to that.
Go ahead.
And if I may, as we look toward the future and potential clinical applications of multiplex immunofluorescence, we all are aware that standardization and reproducibility within an assay across sites, across instruments is absolutely critical. And I think we'll speak about it more tomorrow, digital readiness. But one of the things that Biocare and our partner organizations in spatial biology will look to do is continue to standardize with the reagents that are being used, the SOPs, the instrumentation. It's absolutely critical. And that's where we want to focus by using Encore Pro X, by using the Multiplex and having a standardized workflow with standardized panel. You're getting more reproducibility. You're getting more evidence that this has potential clinical applications paired with digital pathology, which is still being adopted, right? It's absolutely critical that we push for standardization and reproducibility. For us, that's going to be in the form of antibodies, right? The highest quality antibodies, having consistency, reproducibility, everything that goes into the wet lab component is going to influence the ultimate digitization and success of those assays. So that's where we'll continue to focus, and it's kind of the spirit of digital readiness, as we've spoken to.
So that's gonna be a live stream that we're gonna have tomorrow. So at the same time.
Yep.
But I want to take a step back because we have two components of a longer workflow. What is the full workflow done, and who did you decide to partner with, and what do these partners provide, and the whole workflow? How does this support advancing immuno-oncology? And you can totally dissect my question.
That's like 10 questions. That's 10 questions in it.
Yeah, I tend to do this, and then people don't remember what I asked. But let's talk about the workflow and who did you partner with?
Sure. Let me break it down. So. I'm going to go back to what David said. There is multiple pieces of this puzzle. So we have validated a variety of reagent vendors. The main reason for that, there's really no standardization. People are working towards standardization. So in order for us to be fair and equitable for all partners.
It's been a long time since I did IHC, but it was I wouldn't say magic, but you definitely had to pray to like IHC and antibody gods back then. I hope it's better now.
It's better. It's better.
But I think it's not without effort. So that's where
Sure.
the standardization
So
and
that we are working with Biocare, and we also have validated like Avant RX just for other stages and pedophilia as well. But on everything has to eventually getting data is one thing. In multiplex immunofluorescence world, that data has to mean something. Interpreting that data to actionable steps, a person who's doing the work should be able to achieve that. So I think that's where we partner with various image analysis vendors
Okay.
from Visiopharm, Indica Labs, Predix AI Gnostics, to name a few. But all of these partners, like David said, when standardization occurs, it's going to narrow it down to
Yep.
like one partner of reagent side, there's a stainer, there's an imaging, and one partner on the image analysis, because that's the way we should be. We could potentially standardize the panel so that we could get reproducible results. Because in the end, if we are not able to get reproducible results, it's very hard to get this problem. So that's one of the options.
And obviously, like for clinical, that's gonna be so important thing that you're saying. Well, whenever we like get it into the clinical utility, and obviously again plug for the talk 12:30 tomorrow at Chesapeake 79. But that's like even super high bar, right? You may need to talk about regulatory stuff, but even to accelerate research, the simple reproducibility is gonna help so much those people who at the end have to interpret this data. And I always call the vendors the unsung heroes of digital pathology, in this case immuno-oncology, because having something that is deployable, you don't have to like reinvent it every
Sure.
time you try doing it, and helps the researchers, helps the clinicians, helps the doctors advance the space. Um, let's talk about imaging. Let's talk about what this multiplex actually does. How many like
Sure.
markers, infinite
So
or
you are
few?
There you
able
go.
you're able to do up to 10 colors with Multiplex. So again, I'm going to say the sweet spot is anywhere from 4 to 8 biomarkers. It's like the sweet spot according to the work what we have done and what people want to do. So when we built Multiplex, our goal was actually to go after actionable steps what people can take. So in order for us to do that, that was the sweet spot was 10 colors, and you are able to load up to 60 slides at a time. So if you're doing a small clinical trial, or if you're trying to do a smaller cohort, you're able to load the slides at the same time whenever it gets taken from Biocare instrumentation.
How
You can
many at the time?
from the instrumentation, how many can you stay?
36.
36.
Okay.
But on the system, you can load up to 60 slides.
Okay.
And the image format for multiplex immunofluorescence, we have kept it as OME TIFF files. So it enables people to do image analysis. But in terms of utility, we also want to ensure that there's enough speed for the instrumentation, so that
Yes.
speed is very critical in this phase. If it's going to take 4 days or 2 days to scan a slide, it's not user friendly. So we simplified that. Probably one of the fastest multiplex IF systems in the market right now. So our goal is to simplify. So we are able to do 5 colors, 15 by 15 millimeter piece of tissue in 14 minutes.
Okay.
Comparing to a lot of the systems, that's pretty fast, especially if you're able to load 60 slides and go home, come home next morning, you have all your multiplex slides done. So
I know what I wanted to say. So if you look on my left, next to Don, there's this Multiplex image, and I think on this one you guys have 9 colors, or like a lot of colors, and we are waiting for a special gift for you guys. If you are here at SITC, booth 415 today, we're gonna have stickers with these beautiful multiplex images. I just checked when they're going to be delivered, I think, before lunch. So before your lunch break, make sure to come here, and you're going to have a little gift with these beautiful stickers that are waterproof. You can put them on your water bottle and basically be proud of the research that you're doing. Or if you just think it's pretty, which I do mostly H&E, but I think this is pretty, so I'm totally putting it on my water bottle. Tomorrow, David, we're going to be talking about digitally ready. Do you want to give a teaser?
Sure, do that.
Go ahead.
Yeah, so this concept of digital readiness is something that Biocare is very focused on. Obviously, there's a lot of movement, there's a lot of trends and progress in the digital pathology space, of course, multiplex immunofluorescence, but I'll continue to emphasize the importance of everything pre-analytical, pre-digitization. Right? And obviously, there's a lot of exciting things happening through analysis and everything to do with AI algorithms and color unmixing, etc. Biocare is a wet lab organization, and so we want to focus on digital readiness coming off the stainer. It starts with antibodies. All of your reagents use automated staining and choosing it for the job. And we're going to continue to focus on producing digital-ready stains, whether they're chromogenic IHC or multiplex immunofluorescence, to ensure that what goes on to the Multiplex or any digital slide scanner is the best stain slide possible and produces the best data possible for research, translation, and clinical applications. That's digital readiness in a nutshell.
And tomorrow we're going to talk a little bit more about the details. One thing I want to say is that in this space, like often in the general bright digital pathology space, you get pushback that, oh, you can do glass, like digital is nice to have. Here it's not nice to have.
Critical.
Because when, as a human observer, if you're supposed to do something with more than 2 colors at a time, well, 3 you could maybe do in bright field, but I just don't want to do it as a pathologist.
And can't quantify. You certainly can't quantify it, right? No, nothing computational or quantified, right? So I think that's a key element when you talk about quantifying biomarkers, especially a 9-plex, right? It's going to be critical to be digital.
9-plex is beyond my comprehension, but
even 4 colors as a pathologist, it's very hard because it's not just looking at an image. You're trying to understand the neighborhood analysis of what does it mean if 2 cells are there? How many 2 cells are close to each other? When you start putting all this data pipeline, it gets to a point. You need to digitize it. Then you need to have some sort of analyze those data. So it becomes more critical, especially like David said. You go to 9-plex, it's impossible.
Yes, it's impossible. Like I did work with 3 markers. Like the moment you need to do some kind of location, co-localization, proximity, whatever, these are things that you can see but you cannot measure. So not only digitization but also image analysis is a must for this space, and we're going to be exploring this a little bit more. Thank you so much for joining me live today. If you have any questions for tomorrow, let me know in the comments under this live stream, because we can ask them, and you can ask them live as well as we are here, but you may be watching the replay. So if you have any questions, drop a comment. Me and my team are going to be looking at the comments. Tomorrow we can ask the guests, and tomorrow we're going to be streaming from Biocare booth number 717. I'll bring some stickers as well, so come and visit Biocare as well. Thank you so much, David.
Thank you very much.
Thank you so much.
Thanks, Alex. Thank you.